All right. Hello, everyone. It's just after 7 o'clock, so we're going to go ahead and get started. My name is Candace Benson. I am the Outreach and Events Coordinator here at the Virginia Institute of Marine Science, and welcome to the first After Hours of 2022. Thank you all for joining us in person, and thank you to everyone joining us online as well. Tonight's lecture series is made possible by First Advantage Federal Credit Union and Canon Virginia Inc., as well as the many other supporters of VIMS outreach program. So let's get into our talk for this evening. Our lecture tonight is going to be given by Dr. Richard Snyder. He is a professor and the director of the VIMS Eastern Shore Lab in Watchapre, Virginia, a position he's held since 2015. He received his BS degree at the College of Charleston and his PhD at the University of Maryland. Originally, he was from Queen Anne's County, Maryland, and he returned to Delmarva Peninsula after 27 years in La Jolla, California, Saskatoon, Saskatchewan, and mostly Pensacola, Florida. I teased him and said he likes to move to places with very difficult names to pronounce. He's a broadly trained ecologist with experience in basic and applied research in a variety of disciplines, including microbes and their ecology, oceanography, plant community of wetlands and barrier islands, chemical contamination in fish and shellfish, water quality in aquifers and surface waters, and aquaculture. When younger and more able, Dr. Snyder sailed on square-rigged ships and schooners, including the first Pride of Baltimore and races on a Chesapeake Bay log canoe. He currently lives in Craddockville, Virginia, on a small produce and grain farm. And with that, I will turn it over to you, Dr. Snyder.
Great. Well, thank you very much. And I really appreciate this opportunity to talk about one of my favorite critters. So I first personally became aware of bay scallops when I was a very small child. My father's family built a very simple beach cottage on the coast of New Jersey after World War II. And we spent 2 glorious weeks there every year in the summertime. And my mother used to take us to the bayside for the kiddie beach where there were no waves. And we would swim around in the grass beds there. And one day my grandmother was there with us, walking through the grass beds, looking very intently through the grass. And I said, "Grandmother, what are you looking for?" And so, "I'm looking for scallops." I had no idea what she meant, but she was very serious about it. She didn't find any scallops that day. And nor did we find any years following that, wading those grass flats for soft crabs and digging hard clams. Never saw a single bay scallop in Barnegat Bay. And it wasn't until I got to Florida many years later, where they still have a recreational harvest for bay scallops, that I realized why my grandmother was so intent on finding one of those. Because they are amazing critters, amazing just in themselves, and also amazing seafood to consume. So I hope I can tell you a good story tonight. They said bay scallops are intriguing and incredibly appealing. And part of this appeal is just that they're just very symmetrical, right? And it's a symbol that's been used for millennia by various cultures all over the globe. And some of this has to do with things like representing the rays of the setting or the rising sun. And this may come from a Celtic influence. Or in the Christian tradition, it's a symbol of St. James the fisherman. And I just really love this quote here at the bottom that came from this webpage. It says, "The many grooved lines represent the many different spiritual, religious, humanist pathways that lead to the universal center of all life forms, the spirit, the soul." So I think I could just stop right there. We could all go home. That pretty much just captures my talk. And no, let me explain some of the science to you if you'll bear with me. So if you go to the restaurant and you order scallops, those are sea scallops, right? You get a big chunk of adductor muscle meat, and it comes from a pecten about this big. It's an oceanic pecten about this big. These are different. These only get to be about yay big. And the meat is still that round adductor muscle, but it's much sweeter and nuttier than a sea scallop. It is absolutely fabulous. The natural range of these critters is from the cold northeastern Atlantic all the way around the Gulf of Mexico to the Mississippi River Delta. And then there is another species that occupies the western Gulf of Mexico. There are two subspecies of this. So the scientific name is Argopecten irradians for the rays on the shell, right? Irradians. The northern subspecies, Argopecten irradians, is found from, well, Cape Hatteras north. The other subspecies, Argopecten irradians concentricus, is the southern species and typically found south of the Outer Banks and around through the Gulf of Mexico. Now, the Outer Banks are a natural break for a lot of species. We have northern species and southern species, and they tend to follow that break. Although with warming seas and warming summer temperatures, there's a lot of spillover between the Outer Banks and the mouth of the Chesapeake Bay and the eastern shore of Virginia. And we're not really sure which subspecies was in Virginia originally. We suspect that Virginia is a mixing zone. It is for other species like hard clams, a genetic mixing zone. So we suspect that these two subspecies were somehow mixing along our coastline, but we'd really have no proof of that. It's just an assumption. So it is a coastal, high salinity species. So it does not like, we see it a little bit up into the bay mouth, but it is mostly out on the coast. It is behind the barrier islands in the bays, which is how it gets its name of bay scallop. It's predominantly a shallow water, grass bed, coastal bay species. Now, each one of these red squares, we have secured genetic stocks of these organisms from these locations, and we use those at the Eastern Shore Lab for research and for restoration for aquaculture. And I'll be talking about those a little bit later in the class. So we have representatives of the species from the range of its natural occurrence. Now, seagrass is a critical habitat for this critter. And this is its typical attitude in nature. It is we call it epibenthic. It's on the benthos, right? It's sitting on the bottom, but it's mobile. It doesn't stick there. It moves around a lot. And so it will sit, you'll find it sitting like this, filter feeding, just voraciously filter feeding. They're incredibly hungry all the time. We use them for filtration experiments with students because we know they're always going to eat something. And yet they're highly mobile. And I'll show you some of that later too. The critical habitat we have here is eelgrass, Zostera marina, which is a northern grass. There is some down in the Outer Banks, but once you get south of the Outer Banks, then it's haliduli, right? Some other seagrasses, right, that are down there. And in the Gulf of Mexico, Jacksonville, Florida, and down into the Gulf of Mexico, it's turtle grass that's the critical habitat down there, right? Thalassia testudinum. But seagrass is critical for the survival of the species, and that's a key part of the story also. Like many bivalves, it has a planktonic larvae. So the adults will spawn, eggs and sperm released to the water, the eggs are fertilized, develops into a planktonic larvae that will metamorphose after about 10 days and will become a spat, which looks like the adult, only it's very, very small, same as an oyster spat, right? And then as they get bigger, the spat will stick to surfaces, right? They don't permanently stick, but they have means of temporarily sticking themselves on surfaces. And this is where the eelgrass comes in because they climb up on the grass blades and they hide in the junk that's on the grass blades. So that's how they keep from being eaten. But when they get bigger, they drop off the grass blades and then they're sitting on the bottom like that other picture that I showed you. They only live for a year and a half, maybe 2 if they're lucky, which is really unusual for mollusks. Most mollusks are more long-lived than that. And so that's when we're looking at population dynamics of these scallops. That's part of the real difficulty in managing these as a harvestable species is that short lifespan, because one bad year and you can have a population crash, right? And it's hard to recover from that because it is such a short-lived species. So I wanted to just contrast this with another mollusk that you're probably familiar with, hard clams. And look at some of the physics and mechanics of the 2 shell types. So both of these critters are mobile filter feeders. Clams are burying in the sand or the mud, and they can move a little bit, a few inches a day maybe, or maybe a foot if they're really motivated, but not very far, right? They don't go very far. They mostly stay where they are. And they have a very, very heavy shell, right? It's very thick, heavy, smooth, so they can move through the sediment. Scallops are really mobile, and they have a very, very different kind of shell. But these are both filter feeders found in the same habitat. So let's look at the shell. So this graph, here's the weight of the shell and the size of the shell. Now, scallops are only going to live for a year and a half, so they're not going to get as big as clams will. The clams keep growing, and once they get past a certain size, the shell gets massive, 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 very, more than they don't get bigger, they just get heavier, right? Just quickly getting heavier and heavier. So let's focus on this part of the graph right here. That's what this is. And here's the scallop shell. Here's the clam shell. As the shell gets bigger, the increase in weight. If you look at a 45 millimeter shell, which is right here, and here's the width, you break those shells. This is the clam, this is the scallop. At 45 millimeter shell width, the scallop shell is 50% of the weight of the clam shell, right? It's a very, very light shell. Because if you're going to move around, you don't want to be carrying a bunch of weight with you, right? If you're a marathon runner, you don't want to be 300 pounds. So they have a very, very different type of shell. Now, in order to get that lightweight, well, let's look at this. Not only is it lightweight, but it's hydrodynamic. It's got this beautiful curved shape. The old shipwrights used to call that a mackerel shape, right? And they made their boats look like mackerels because they were fast. That hydrodynamic shape to it. And they also have slots in the back of the shell. Scallops never completely close. Clams clam up, right? Throw a clam on the sidewalk, hot day, it might even sit there for a couple of days and still be alive. It can close up and be watertight. Temperature is probably worse than dehydration for a clam. These guys dry out because they can't close watertight. And that's a real problem when we're trying to grow these for aquaculture or harvest and getting them to market. We've got a limited market window to move these before they dry out and die. Right? Clams, you buy clams, they're dry, right? When you buy them from the store, they're dry in a bag. Can't do that with scallops. It's a different beast. But those slots are there because it's jet propulsion. When they slap their shells close, the water's going to squirt out those slots, and that's what moves them. That's how they swim, right? That's how they swim. So they've got jet propulsion. It's a hydrodynamic shape. They've got wings on the back of the shell. And if you look at the ridging, right, the scalloped ridges and valleys, it's strength without the weight. It's like corrugated tin on a roof, right? It's strength without weight, but those ridges also reduce drag and increase lift for swimming. So everything about the shell is adapted for motility, and that all equals the ability to swim. And this is what it looks like. This is somebody swimming through the grass, comes across a scallop, scallop jumps up, and it swims away. Want to see that again? This is really cool. Who would ever thought, right? Look at that. That's unbelievable. So they can move pretty considerable distances. Now, what we don't know is whether there's any schooling behavior, dare I say a social interaction, maybe not. Scientists don't like to use that word a lot, but is there any kind of interaction, aggregation, how does this affect the distribution in the habitat? We really don't know. What we really need to do is put some little mini cameras on these scallops so we can see where they're going, or GPS trackers or something, and figure out, because we really don't know a lot about their movements. We know that they do move, and sometimes considerable distances, but we really don't know why. So what about the eyes, right? They have these beautiful blue eyes, unbelievable color. The photograph really doesn't do it justice. These are incredibly complex eyes. These are mollusks. These are bivalves. And scallops have a much better developed nervous system than oysters or clams, but they're still mollusks. And yet these eyes are incredibly complex, and all scallops have these. This Dan Spicer did his dissertation work down at Duke on different species of scallops and was interested in the evolutionary history of them. But do you know what a mirror reflex lens is? So a telephoto lens for a camera, right, is a series of glass, and it magnifies the image to film or electronic pad or whatever it is you're going to sense the picture on. And in order to increase the magnification, you increase the focal length. That's why at the sporting events, you see these guys with great big lenses, right? It's a long focal length. Well, you can get the same focal length in a short lens if you use a series of mirrors to bounce the light back and forth to increase the focal length. That's what these guys are doing. They've got a mirror reflex eye. And not only is it a mirror reflex eye, but the light comes through, see a diagram at the bottom, the light comes through the lens and hits the retina tissue, bounces off a mirror in the back, and this is a focal mirror, and then is focused back to the retina. What kind of an image is that? What does that look like? We have no idea, right? But it's got to be a complex image of some sort, or some sensing means of the environment. We have no idea exactly how that works. We also don't, they have no brain, right? They're lucky to have, a bunch of nerve cells stuck together in a ganglion somewhere that doesn't really constitute a brain. How do they transmit visual information? Well, we've got a whole chunk of our brain to deal with visual stuff. These guys don't. How do they translate visual information into behavior? We have no idea. No idea. We do suspect that for a long time that it was just light and shadow. So like the scallop that jumps up and swims away, he sees the shadow of the cameraman coming. Or a fish, a skate skates love scallops, or a crab, or some predator coming. You'll see the shadow and it can jump away. And they've done experiments where you put a scallop next to the grass bed and they can sense the darkness of the grass bed and swim towards it, right, to get to their preferred habitat. But what Dan did was take a scallop, glue it to a piece of PVC pipe, plastic pipe, in an aquarium with filtered water, clear water, right next to the glass. And on the other side of the glass, he played videos to the scallop. Videos of particles going by. These are filter feeders. They're feed on particles. So particles of different sizes, particles of different densities, different speeds, and some combinations of those, the scallops would open up and the feeding tentacles would go out and they'd get really excited. You could see them, they're really happy about some of those images. No other stimulation other than what they saw through the glass. So they can see a heck of a lot better than we think they can. But what does that look like and how are they responding to that? We have no clue how all this works. But it's incredible, it's amazing, right? How amazing is that? But a very, very incredibly developed and elaborate eye that these guys have. So what about harvest? People have been eating scallops. Probably is the first hominoid that ever walked in a shallow grass bed could pick them up and eat them, right? We know the Native Americans ate them, and there's been a substantial harvest, especially in New England, has been the most sustained harvest for these. Cat boats were very common, this type of boat with a single mast, forward mast rig, dragging a dredge for scallops or a rode for scallops. Now everything is done with motorized vessels, right? And you can see the harvest intensity up here. So this is 1970s and 80s, but now the modern boats, this is 2007. So the typical arrangement is still pretty much the same, just two dredges off the stern and you pull it up and you sort out one pair while the other pair are running, and very, very efficient way to harvest these critters. And it has been a big harvest for a while until 1980. So here's bushels. This is the Atlantic coast from Massachusetts to North Carolina. And you can see the harvest is going along pretty well. So we get to the 1980s, and then the harvest plummet. And scallops have been in trouble up and down the East Coast ever since. A lot of this has been ascribed to habitat degradation, loss of seagrass, a critical habitat, harmful algal blooms in some cases, but they just have been in trouble and not recovering up and down the East Coast. In New England, they still have harvests. Occasionally they've had collapses. New York, Martha's Vineyard, Nantucket, a lot of these places have hatcheries and they augment the natural population from the hatcheries, and they still have commercial harvest in these areas, but nothing like it was prior to, right? And not only is that across that's the broader picture across the Atlantic coast, right? And there are more localized effects. So here's where my grandmother was looking for scallops in Barnegat Bay, and they were basically gone by that point, right? She couldn't find any. They were no longer there. I never saw one after that. Gone. One little data point from Delaware Bay, and no other records again. North Carolina, the harvest continued for a while, and they've not opened a harvest in North Carolina for many years now. They're still there, but the numbers are not high enough to sustain a harvest. And they can't figure out why. We really can't figure out why they're not bounding back. Habitat is certainly a key issue, but it does not seem to be the only issue. It's a mystery to us. So what about Virginia? There was a commercial harvest in Virginia out in the coastal bays. Again, this is a saltwater species, an ocean salt species. So they were found as far up as Cherrystone Creek above Cape Charles, and probably a little further, but I hear, we have somebody at the lab who's from Tangier, and we hear reports from Tangier every once in a while that somebody says they found a scallop up in Tangier, but that's a long way from the ocean for a scallop to get. It's possible, but 25 parts per thousand is about the limit. But in 1923, the Commonwealth set up public harvest areas for scallops, and there was a commercial harvest. And here you can see the harvest in kilograms and the year. And here's where that regulation, the regulations were set here. This is a scallop dredge. It's now owned by the Nature Conservancy. Actually, it's owned by Bo Lusk, who works for the Nature Conservancy. I think he just keeps it at the Nature Conservancy. Although, its ownership, I think it's Bo's, I think. But it comes from the 1920s and was found in somebody's barn. And, that was used back in the 20s for scallop harvest in the coast of Virginia, typical scallop dredge. So this graph shows you the scallop harvest here and the clam harvest. And we won't worry about the oysters right now. So right about here, when the regulations were put in, you see this exponential rise in harvest. And apparently over harvest because things started to drop after that. This is right about the time when people started putting motors in boats, right? Single cylinder putt-putt motor in a boat, and they could be much more efficient than trying to do this under sail or oar. After that peak harvest, they started to drop, and then we get to this dash line right here. This is the 1933 hurricane, which devastated the Eastern Shore and destroyed all the grass beds and all the coastal bays. There's still grass up in Chincoteague, but the grass from all the coastal bays was smothered. The sediment covered it up, and it didn't come back. There were little patches of it here and there, but it just never recovered. And so VIMS started a restoration program. J.J. Orth, who recently retired from VIMS, and now Chris Patrick, who has taken his place, have been restoring seagrasses to the coastal bays out here. Oyster is like right here. Oyster, Virginia is right there. And you can see we're now to about 9,000 acres of grass that we've recovered back in those coastal bays. And about 2009, Mark Luckenbach, who was then the director of ESL, at about 6,000 acres of seagrass, he went down to North Carolina, so we're going to put scallops back into the grass beds. The scallops had been extinct, locally extinct from Virginia for nearly 80 years, right? Just gone and never came back. Mark went down to North Carolina, brought scallops back to the Eastern Shore Lab, spawned and reared them there. And we have been rearing and releasing scallops to the grass beds ever since. And we are trying to get a natural wild population restored to the coastal bays. And I'll get into that here. Well, let me tell you how we do this first. This is a ripe bay scallop. So we keep our scallops in cages so we don't have to go round them up when we want to bring them in to spawn. So we keep them in cages, and they're filter feeding in the cages, and they will ripen when they're ready to spawn, and we keep track of how ripe they're getting and bring them in. Usually late April, May, we'll bring them into the hatchery, and they are both male and female, so they are hermaphrodites. And this is the eggs right here. And this white stripe is the sperm here. So they will produce both eggs and sperm, but not at the same time. They do it sequentially. And by doing it sequentially, there's very little danger of them or less danger of them fertilizing themselves, right? Selfing, which would reduce genetic variation. It's not adaptive, right? It just works against their survival. But you can see those, again, those beautiful blue eyes around the mantle, but they are both males and females. There's a significant spring spawn, and we also get a fall spawn where we are. Up north, there's usually just one spawn. Down south, the further south you go, the more strong the 2 spawns a year occurs. But we get a major spring spawn and a little weaker fall spawn. So we bring the scallops into the hatchery. We grow algae food for their filter feeders, right? As larvae and as adults. So we grow algae, cultured algae for them to feed on as larvae. And they are conditioned, grown, and reared in tanks in the hatchery. So typically they'll come in, we'll bring them in, put them in a spawning table like this, right? And we'll warm them up a little bit, bring up the temperature a little bit, light some candles, put on some music, and then we get this. So watch right over here. See that? That scallop just spit out an enormous number of eggs. So the eggs and the sperm are released into the spawning table. And after either we're ready to go home or they're done, we'll drain that water down onto a sieve and we'll collect all the eggs, rinse them, clean them. And then those eggs will be resuspended in a large tank. And we'll put cultured algae in there, suspend cultured algae in there, and they will hatch. And those larvae will grow in that cultured algae for 10 days in the planktonic phase. And, it looks like this. So they start out as an egg, and they gradually get bigger. And we call this an eyed larvae because you get that dark mass that grows inside. It's not a true eye. The eyes are around the edge. But that dark mass ends up, we call it an eyed larvae. And at 10 days, they'll get to a point where we call them petavillager. And the petavillager, petty foot, right? Petty foot. The petavillager has a foot. And they use it. And they move around quite a bit. And what we see with these larvae at ESL is we put them into, now we take them out of the hatchery. They're ready to settle. They're going to the bottom, right? And they're going to use that foot to move around. Now, classically, everybody thinks that these guys land on the seagrass blades and that's where they stay. They stick themselves with, they actually have byssal threads like a mussel too. They use the foot and the byssal threads. They stick onto stuff and then crawl around, change their position. What we find in our nursery tables is a lot of them land directly on the bottom and then crawl up on structure. So in nature, we're not sure if they're landing on the grass blades. We know some of them are. But we're wondering if a lot of them just land to the bottom and then crawl up the grass blades, right? Because that's what they're doing in our nursery system. And it may be a condition of the artificiality of our nursery system, but it may also reflect a behavior that they're doing in nature. That's something we would really like to figure out. So after a lot of work in the hatchery, this is what 2.4 million petavillagers look like. And you put all your eggs in one basket and you hope they survive. But it's kind of disheartening sometimes what you boil all your work down to. We keep track of survival. So here in the hatchery, these are the fertilized eggs. Not all of them hatch successfully in the larvae. Once they do, then we lose less of them over time. And you see that here with survival. This is the number of larvae. This is survival. So there's very low survival of the fertilized eggs here. But once they hatch, survival is pretty good in the hatchery. And then the petavillagers are taken out to the nursery system. And that's where we start to really lose efficiency. And so a lot of our research and culture has been out here, right? This transition period and in the nursery itself to try to improve the efficiency there. So this is the nursery setup. And there's Darren, who's in charge of the nursery, as usual, doing the work while I'm standing around talking. And you see these slats in here. These tables are fed with water straight from Watch Sprig Channel. So at this point, we're switching them from cultured algae to natural plankton. And the pettivillagers are loaded into, larvae are loaded into these tables. And again, a lot of them are falling to the bottom, and then they climb up on the slats, right? They like to be up, and that up is into the seagrass blades where they can get food and find shelter. It's a lot of labor. Fortunately, we have interns who come help us every summer, and these tables have to be cleaned every day. And once they get a little bigger, you start seeing all the scallops, all these black things. That's all baby scallops on here. But when we first put them out there, they're 0.2 millimeters. So everything we do in the hatchery and the early part of the nursery is all by faith and microscope, because you can't see them with your eye, right? You hope they're there. And we check them with a microscope every once in a while just to make sure. But it's not until we start to see them that we really get really excited, because then we can see how many are actually there. So our nursery production, our hatchery and nursery production by year has been pretty sporadic. Not really sure what happened in 2016 and 2018. 2018, at least the scallops just did not condition well. And we're not really sure why, something in the environment. And again, they only live for a year and a half, one bad year, and the population could go. So this is a little disturbing, but it is odd that there's this sort of alternate, every other year we've had a low year, except when you look at 2020, the reason we had a low year is we had to cut back production because we couldn't hire interns to help us. So that was not anything to do with the scallops, that was just lack of labor. But you look at 2021, you look at the high bars here, or even the 2016, 17, if we take 18 out of this, because we know they didn't ripen well, 16, 17, 19, and 21, we're increasing our production ability at ESL, right? Darian and Reba, who run the nursery and the hatchery, doubled their production from one year to the next. Unbelievable. Just the work these two girls are doing out there is phenomenal. And so that's 1.4 million scallops that we had available for restoration and aquaculture. And this is Darian and Reba. That's the dream team out at ESL that does the hatchery and the nursery work. So once they're 5 millimeters, then we can put them into a 3 millimeter mesh bag and not lose them. And we'll put them in a mesh bag and put them in an oyster culture cage. These cages you see are typical for oyster bottom culture. You may have seen them in a different after hours talk. The mesh bags go in there with the scallops, and as they grow, we'll move them to bigger and bigger meshes, because the mesh allows the food to go through, and the bigger the mesh, the more food they can get. So we keep sorting them, grading them, and moving up to larger bag sizes, different mesh sizes. And ultimately, they get big enough so that they can be free in that 1 inch square mesh wire, right? And then they're free in the cages. Well, free in the cages. How can you be free in a cage? They're no longer in a bag in the cage, right? But they're still in there in fairly high density. And one thing we notice is that the scallops that are confined tend to get fouled shells. There's a table of scallop shells out here, and these are all cultured shells that we had. And you can see some of the fouling on them. If you'd like to take a scallop shell or 2 or 3 or 4 home to kids, grandkids, or yourselves, please help yourself. They're on the table outside. But the scallops that are in these cages tend to get fouled. The ones we collect out of the grass beds, absolutely clean. And it's probably because they're moving around, but also because they're sitting on the bottom and they can smother whatever's growing on their shell, right? It's a really dramatic difference between the cultured ones and the uncultured ones. But the cages have to be swapped out. They've got to be clean. They've got to be hauled up. They've got to be, split, sorted. There's a lot of labor involved in aquaculture. And then occasionally you run into somebody like that guy up there in the right corner. You open up the cage, there's a giant fat octopus in a cage full of empty shells. And he was having a field day because the sharks couldn't get him. He was in a cage and he had all the luscious scallops that he could possibly feed on. And fortunately, that doesn't happen too often, but occasionally it does. So we release a lot of bay scallops to the grass beds. And we do this in all stages. We release pettivillager larvae, the ones that are ready to settle. We release them as juveniles and adults, and we're hedging our bets, right? We don't know if we drop those pettivillagers out there. There's other stuff in the plankton that eat stuff like this. Everything that we drop out there is immediately going to be food for somebody, and so we've got to play a numbers game. We want to release enough so that we're saturating any predators that might eat them, and hopefully some of those are surviving. So we release all the stages that we can, and as often as we can. We also do what we call in situ spawning, so in place spawning, in situ spawning. So the scallops we have out there right now are, the density is so low that they can't possibly mate. They can't find each other, right? They're too far apart. So they release eggs and sperm, and the eggs and sperm just get diluted and washed away, and they never fertilize. By keeping them in cages, we've got a higher density of scallops. When they release eggs and sperm, they can fertilize, and we can produce larvae out of the cage scallops to seed the grass bed. And that's another strategy that we're using for this restoration work. We have an annual survey to keep track of the population out there. We literally take a PVC square that's 1 meter square, and we lay it on the bottom on the grass, and with our hands go through and pick up all the scallops we can find. And it's a lot of bottom, because we hit 16,000 of those square meters. We've actually cut this back the last couple of years, but a lot of bottom that we cover by hand. And it's a fairly intensive effort, 2 or 3 days, 5 boats, 4 or 5 persons per boat. And it involves ESL and people here at VIMS, Chris Patrick's lab here at VIMS, and we get the Nature Conservancy to help us too. So we try to get an idea of where the scallops are found, how many are out there, what the sizes are. The size spectrum is important, right? If they're all big ones, that means there's no little ones for next year. If they're all little ones, what happened to the big ones? So we want to keep track of the population size as numbers and also as individuals. We take DNA samples. We want to keep track of the genetics. And we collect all those wild scallops and use them as broodstock for the restoration, right? So we try to cycle back the scallops from the grass beds to the grass beds. So this is what the data looks like for that. And it really is kind of noisy. So here's those 2 years with low hatchery production, coincided with 2 years where the population dropped. And again, you know, it's not good science having 2 data points, so it's hard to really say anything about that. It just seems maybe coincidental. But this was 2020 with COVID, and we had very low hatchery production, nursery production, because we didn't have the labor to do it. And yet the population continued to increase right after that. So that's pretty encouraging to me that we didn't release many scallops that year, but the year following, the numbers were higher. So either the spawning cages or the releases or that we've done before, or the scallops that are out there are finding each other finally. And as the density goes up, there's some critical threshold that we need to get to so that it's sustainable, right? So they can keep reproducing themselves and we can stop growing and releasing them. We don't know where that is yet, right? We don't know where that is yet, but we hope that this trend continues and we get to that point. So, you know, I'm not holding my breath, but this coming year, it'll be really interesting to see if we're up here or if we're back down here, right? It's really hard to tell. But you get an idea of you know, all the sample points we hit. It's a fairly exhaustive survey through the existing grass beds. Now, why do we get this data? So down in Florida, where they have a recreational harvest for these, which is quite fun, they manage it and do pretty much the same thing. They get census data. They find out how many scallops are all in different areas where they're open for harvest. And they use these criteria. So if you look at what they consider collapsed, vulnerable, stable, and sustainable, they won't open a harvest unless they've got more than 1 scallop per meter squared, right? You're looking at a meter square, 1 scallop in every meter square on a grass bed. That's a lot of scallops. And I can tell you from scalloping in Florida, I dove down in 15 feet of water and picked up 4 of them on one dive. I mean, just go down, boom, 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 they're just right there. Amazing density of the critters. We're nowhere near that, right? Look, where we are, we're down, we're 0.01 to 0.1. Here's 0.01 right here. So we were in a collapsed state here below 0.01, and we are still in a vulnerable state according to Florida criteria, right? We're not even close to a stable or a sustainable state, but that's our goal, right? We want to get this population up to that point, and boy, who knows? Maybe even a recreational harvest, probably after I retire. I don't know. It could be a while. Could be, you know, if this thing takes off, who knows? We hit that. Where's that threshold? We haven't found it yet where they take off. And Yeah, so we're just hoping that we get there. Jan McDowell here at VIMS is helping us with the genetics. And I don't have New York genetics in here. And this was just the samples from the scallops we brought up from Florida. So you can see here's our brood stock and scallops from North Carolina that all cluster together. The Florida scallops are very, very different genetically. And I think if we bring in the New York data, they'll also be very different. I don't know where on this graph they will show up, but I'm suspecting they will cluster away from this and away from this. And if you take this part out, the Florida scallops out, and just look at these and blow that up, they're pretty much centered around the North Carolina scallops, right? So our latest data is getting more and more focused around North Carolina scallops. And those are the ones that we brought in the most recently, and the ones that the DNA, the alleles, the genes from these critters have spread into the population, and now they look very much like North Carolina scallops. What's going to happen when the Florida genes get out there, when the New York genes get out there? Which of those genes are going to end up in our population, and which ones get tossed out, right? They don't survive. That's one of the things that we're looking at going to the future, is how the genetics change in this restoration process. So what about aquaculture? So even though scallops were absent from the coastal Virginia habitats for 80 years, Mike Castagna here on the left, the first director of ESL, he and his crew were working on aquaculture of bay scallops back in the 60s and 70s. And Mike Osterling was out at ESL in the 80s and was working on bay scallop culture. Mike Osterling even had market connections to chefs in Washington, D.C., but he just couldn't hit a price point where the aquaculture was profitable, right? It costs him more to grow them than anybody was willing to pay for them. That's changing. If you look at this graph, there's pounds. There's that same graph with a drop in the 80s, right? Goes across, drops in the 80s. So this is the harvest data. But then look at the price per pound. Population crashes, they become more rare. Supply and demand, price goes up. And now we're getting up into price ranges where aquaculture may be profitable for producing these critters because the wild harvests are disappearing, right? And just to make this more real, 2019, I was in a grocery store in Virginia Beach, and I don't know how they get away with fresh for everybody and fresh exclamation point with bay scallops flown in from China, but $7 a pound for bay scallop meats. That same year, the population up in Martha's Vineyard collapsed. People were willing to pay $30 a pound for local, right? And want stuff from China. But this is a big issue, overseas imports, especially Chinese imports. China has the labor force to do this. And these are aquacultured scallops. These are wild harvests. But when the wild harvest collapsed, people are still willing to pay it. This is about $1 a scallop is what that boils down to, right? That's clearly an aquaculture range, but this is an exception. So we take some of our scallop seeds, the little 5 millimeter ones, and we send them out like 10,000. We'll send them out to a commercial grower, or 5,000, or whatever they want to handle. And we'll try to get commercial growers to grow them, right? Get them interested in it and try to find them a market. When we do that and compare it to our cage results, here's a couple of commercial results from 2020. They're getting pretty much the same results that we are working at research scale. Now, we baby them along at research scale because we don't want to lose them. And to find out that the commercial guys can be a little more rough with them and not pay as close attention to them and get similar results is pretty encouraging to us. But what's more encouraging is we assisted a man fresh out of the military. He decided he wanted to start a scallop-only farm on the Eastern Shore of Virginia. Actually, he tried to do it in Maryland, and the bureaucratic red tape for aquaculture in Maryland was too much, and he moved just south of the border into Virginia to do it. And we helped him get started. We supplied some seed to him. He got seed from a commercial outfit in New York, actually. And he's using these, they're called lantern nets. So it's a series of stacked nets. And you can run this in deep water. You just have a buoyed line that runs across, and you can suspend them across. So he was able to successfully partner with a hydroponics guy who's growing vegetables in a greenhouse. And with the scallops and the fresh vegetables, and hitting the local markets. He made a successful go of it for 1 year. We're hoping he's going to repeat this, and we're going to be there to try to help him if he can. But I think we're at the point now where we've sort of broken this price threshold for aquaculture and bay scallops, and that may be part of the future. We're hoping that it is, and we're there to help see it go. So these are our costs up here. And this is from 2019. And remember, we doubled our production from 2019 to 2020. So the seed cost would be half of that. But this is the real kicker. You know, when you get out to the grow out, once you get to 5 millimeters, you still got to get them to market size and make money on it, right? So you start losing money at different points in this. But even so, at our research scale, we were about 30 cents to 50 cents a scallop. And certainly this guy out here with his lantern net and his scallop farm, he's doing a heck of a lot better than that. And hopefully he's making some money at it. We'll see how successful it is going forward. So that's basically my scallop story. But before I quit, I wanted just to show you where we are. So we're over here at Foster Point, and Eastern Shore Lab is over here in the little town of Watchprig. And this is Watchprig Inlet. And we are located at the north end of town. So VIMS has been working over on the Eastern Shore. There was a little field station there in 1941. They were doing aquaculture on salt marsh mussels. So aquaculture has been a long legacy of the Eastern Shore work. VIMS was officially established in 1960, but now we are completely rebuilding the lab. There'll be 6 new buildings coming online this year. And please come and visit. We have an annual public open house, 3rd Saturday afternoon every September, except the last 2. So hopefully this year, please come visit. Come over to the Eastern Shore and come visit the lab. It is usually a great fun, a great time. And if you have any desire to see other things on the Eastern Shore, you know, drop me a line. I'll be glad to give you a list of things that you could do. So clearly these things do not happen in a vacuum. And I'm certain that I've left people off of this list, but there's an enormous number of people and lots of funding agencies that have made this work possible, and including private donors. And we're very grateful to all of it for making this all happen. So thank you very much. Glad to try to answer any questions if you have.
I saw pictures of the scallops in the shell, and I saw pictures of them in the grocery store. What's the step in between? Are they like shucking like they do with oysters, or is that a problem to find people to do that? And is that
yeah? So they are taking the adductor muscle, like when you go to the restaurant and get a scallop, that's coming from this critter right here.
Right.
This is an ocean scallop, and you need to talk to David Rudders here at VIMS, who works on sea scallops. Right. So the bay scallop is very similar, has a single adductor muscle in it. And the traditional market is to shuck them, pull that muscle out. And that's all that you're buying is just that adductor muscle, just like a sea scallop. But the whole thing is edible, and they're actually quite tasty. And that's what we're trying to do, is market it as a whole organism product.
So is there a market
great with linguine, a little olive oil and garlic, and you know.
So is there a market for the rest of the scallop now?
No, there isn't right now. So I mean, there is. And there's actually a French recipe out there from a long time ago where they ate the whole thing. So it's been known, but a lot of people don't like the look of it, right? It turns some people off. And there's one guy up in Martha's Vineyard who's telling me, I asked him about eating the whole thing. He said, oh, no, I don't do that. And I said, why? And he said, well, because I see the cats that are going over where they're shucking all these and throwing all the everything but the muscle. And he said, all the cats are missing their ears. And I'm not going to eat them. So I think the cats are missing their ears because they're fighting each other, or the birds are pecking them or something. I don't think it has anything to do with the scallops. So yeah, you can eat the whole thing, and it's quite delicious. Raw, cooked, either.
I've never seen a whole scallop.
No, you won't. No, you won't. No. And even in Florida, where it's a recreational harvest, most everybody shucks them and cleans them and just takes the adductor muscle. Yeah. But it is possible to eat the whole thing. Thank you. Yes.
Hi, thank you. I have a question. You stated in the presentation that you take DNA samples from the scallops. Can you elaborate on that process? Like when does that occur? Like what stage are they in, and how do you do it?
Yeah, so scallops are easier to open than clams. You got to pry a clam open. The scallops will actually sit there and clap. And they'll clap in your hand. So if you're quick, they clap, you grab them and the shells are open and you can cut a little piece of the mantle tissue and it'll regrow. So you don't have to kill them. So you take a little piece of the mantle tissue and do a DNA extraction on that and do a PCR reaction and sequence it. And they look for, it's called SNPs. So if you've got, every gene has different forms, we call them alleles. And so if you line up all the alleles for a single gene, you can see which of the DNA code is different between the different alleles. And those locations where they differ a lot, those are called the single nucleotide polymorphisms, or SNPs. And it allows us to distinguish between the different forms of the genes. So these are, that graph was compiling all those differences in the genes of these scallops and looking for, which ones are genetically different than the others. Right? There's also a mitochondrial sequence that's used, but this was with genomic DNA. Yeah. Does that answer your question? That gets a little bit complicated.
No, but I just thought it was really, I found it interesting because there's just things you don't think about. But
yeah.
But is there a particular, you take the samples when they're more mature as opposed to when they're young, or?
Yeah, when they're adults.
When they're adults, okay.
Yeah, and so the reason we're doing this, in a lot of conservation biology, they're very worried about population genetics. And there's a really good story published on this for Florida panthers years ago. So there are a little remnant population of panthers in the southern Florida. And panthers used to be all the way across Texas to Florida all the way. So this population got isolated, highly inbred, and started to deteriorate. They brought panthers in from Texas and introduced genes back into the population. And a lot of the physiological problems of those panthers went away. And they started reproducing again. That was one of my concerns when I got here, was how many years had we been raising? We started with like a couple hundred scallops and started this huge population from a couple hundred scallops. How much inbreeding was there? Lack of genetic diversity. That's why we're bringing in scallops from elsewhere, to try to bolster the genetic diversity and the genetic sustainability of that population, right? To make it a more healthy genetic population. So we want to track that. How genetically diverse is the population?
Sorry. So I noticed on the graph that you mentioned Florida. So are you concerned about changing the diversity to an extreme, or just?
Yeah, so there's inbreeding depression, right? When things get too inbred. There's also outbreeding depression. So is there a possibility we take Florida scallops, mix Florida genes into the population, it makes them less well adapted to where we are. But water's warming up. I'm thinking we bring scallop from Florida. They're used to really hot water temperatures, and they may help this population survive warming waters. So we've already lost blue mussels out at Virginia coast, right? It's getting too warm. They're moving north. We don't see them too much anymore, every once in a while, but it's too warm. They all die. Surf clams are offshore and moving north because the waters are warming. Shrimp are showing up. So things are changing. So how do we get the genetics into this population to allow them to adapt to that change? Now, bringing New York scallops down, that might be the opposite, but we also have sea ice and freezing temperatures in the winter. So our scallops are in kind of a unique situation where they've got to deal with both the cold and the warm. And so I'm hedging my bets that bringing in these genes, that the environment will select for the ones that are best for survival in this location, and all the rest will disappear. That's the goal.
My question is, I was wondering whether there was any evolutionary benefit to having a changing sexuality, like for the muscle.
So scallops are, scallops are always both sexes, and things like oysters are not. So oysters are, we call it a protandrous hermaphrodite, proto-andros, first male. Mm-hmm. So all the little ones are males, all the older ones are females. Mm-hmm. Now that makes a great deal of sense for energetic purposes, because as you get bigger, you've got more energy to put into eggs. Eggs are more expensive than sperm to make. So it makes sense to have the small ones, males, they can match number for number, sperm for egg, with the bigger organism that can put all that energy, that lipid energy into the eggs and match what a smaller one can make for just sperm, which, packet of DNA. So that makes evolutionary sense to do. Why these guys do both? I don't have any clue. Nope.
And also, what is involved in cleaning a nursery table? It seemed like a fairly involved process.
Yeah, so a lot of it is the sediment load in the natural water. So it is mostly tilting the table up and rinsing all the sediment and the mud from the bottom of the table out and collecting all the scallops that try to escape on a sieve and throw them back onto the table. And it takes about an hour per table or less. Darian's better at it than I am. Probably takes her a lot less than an hour.
Okay, thank you.
Yeah.
Hi, so I'm representing the online group of viewers. We're getting a lot of great questions. I had a few questions wondering if there are any viral or bacterial pathogens that you're finding with the scallops.
Yeah, excellent question. We don't know. Ryan Carnegie at VIMS checks our scallops periodically, and I do know that there's a bacterium that attacks the hinges and dissolves the hinge tissue. And sometimes in our older scallops, they call it a shell slip, right? The hinge dissolves and then it disarticulates. And that's kind of the end of the scallop. So I don't know how prevalent that bacterium is and if that's the cause of why we see so much shell slip, but that's certainly one that's on my radar. Almost everything's got parasites, and I suspect there are some parasites out there in the scallops. We don't really have a good handle on that. So the quick answer is there's a lot we don't know. Don't let anybody ever tell you we know everything in science. It's just not so.
Yeah, let's do one last question before we wrap it up.
Hi, I was wondering, has anyone looked at or done any studies on the shell size due to ocean acidification?
No, we have not looked at that with bay scallops. And it is something that's on a lot of people's radar, especially the hatcheries. And a lot of the hatcheries will adjust pH and alkalinity of the production waters to make sure that they've got enough calcium carbonate saturation in their water. We have not had a problem with that so far in Watch Prig. It doesn't mean that we won't. I can tell you that Watch Prig channel water has increased 2 degrees centigrade in the last 50 years. So we're certainly seeing temperature increases there. pH is not one. pH is something that marine biologists never measured. The ocean's always the same, and we never worried about it. In fact, if you bought a water quality meter for the ocean, it never had a pH meter on it. Now they all do. It's not something that we have a long record of. And I don't know, there are people working on oysters. I don't know of anyone working on scallop larvae with that regard. It's a very good question, and there may be people working on it. I'm just not aware of it. So far, it has not been an issue for us. One more?
I'm just curious about microplastics. Have you seen a lot of that in
So,
the scallops?
no, we have not. But where we are out on the ocean coast, I mean, we see plastics wash up on the beach, but nothing like Bayside or in the bay. It is really a remarkably clean environment. In fact, it's hard to find litter, bottles, any stuff out in the marshes. It's just, there's just nothing out there but nature.
Okay, I was really thinking more about in the animal.
Yeah,
In the animal
I
itself.
don't think that anybody has looked. It's certainly something that might be the case in any filter feeder. But again, I think the abundance of microplastics out in the coastal environment is going to be a lot less than the Chesapeake Bay. A lot of your microplastics, like the hand cleaner and the face cleaners with all the little particles in them, we don't have any sewage going out to seaside. We have some septic tank drainage, but not much of it gets to the seaside. So it's not like here in the Bay where we've got all these major rivers coming out of urban areas and sewage treatment effluent and stormwater. We just don't have the stormwater litter pollute problem, stormwater plastic problem. Most of our rivers are extremely short. Runoff is, in fact, ocean water comes right up to the land. There's very little freshwater runoff on the eastern shore. So the prevalence of plastic particles is much, much less.
Okay.
But it's not something that we've measured. I'm just speculating on that. Yeah.
Thank you.
Yes.
Awesome. Thanks. Those were great questions. Thank you, Dr. Snyder, for
Thank
joining
you.
us.
Thank you all for your attention. Nobody fell asleep.
Thank you to our volunteers who came out. Thank you to everyone who came out to our first after hours of 2022. And thank you to everyone who joined online. And we look forward to seeing you in March. Have a great night, everyone.
Thank you all.